So thank you all for joining us. Today we're talking to Dr. Mohamed Saad from the University of Adelaide. We've had a couple of questions come in via email, which is great. But if you do have any questions during the conversation, please just drop them into the chat box. You can find that at the bottom of your screen there. And I will weave those into the conversation as I'm talking to Dr. Mohamed as well. The session is 45 minutes long. And we look forward to learning about the brilliant work that one of our newly funded researchers is doing. So let's kick off. I mean, you know that moment when you discover something that could change lives? Well, that's exactly what happened in Dr. Mohamed's lab. Thanks to his Cure Cancer grant, he and his team have uncovered what might be a crucial off switch for small cell lung cancer, a specific enzyme that appears in unusually high levels in lung cancer tumour samples. This discovery could be the key to slowing down one of the most aggressive forms of lung cancer. So, as we mark Lung Cancer Awareness Month, there couldn't be a better time to learn about this groundbreaking work that he is bringing hope to patients and loved ones all around the world. Pioneering research like Dr. Mohamed's starts with support from people like you. If you are in a position to help fuel the next breakthrough, which could affect your family or other families all around the world, please do consider donating via the link in the chat or the QR code on screen if you're watching this from our YouTube channel. As I've said at the beginning, please drop your questions into the chat, and I'd love to ask those to Dr. Mohamed as we go through this conversation. So, without further ado, welcome to Lunch and Learn, Dr. Mohamed.
Thank you. Thanks, Nikki, for inviting me, and thanks for everyone joining today.
It's an absolute pleasure to have you here, and we're really excited to learn about your groundbreaking research. Before we get there, many of the people on the call might not know about you, Mohamed, as a person. Could you give us a bit of background about yourself and how you became interested in research?
Sure, no problem. So my background is Egyptian. I grew up in Egypt. In a beautiful city on the Mediterranean Sea called Alexandria. I did a Bachelor of Pharmacy, so I'm a pharmacist by training. And then I did a Master's of Biochemistry to understand how molecules work, basically, in our body. And then I moved to Melbourne to do my PhD at Monash University, where I was interested in the major subtype of lung cancer. It's called non-small cell lung cancer. And then I worked there for 5 years after my PhD, and then I moved to University of Adelaide as a senior scientist at the beginning of this year. And thanks to Cure Cancer funds, I'm able now to study the most aggressive type of lung cancer, which is small cell lung cancer.
Yeah, absolutely. And obviously lung cancer remains one of the biggest, sadly, killers globally.
Absolutely.
Yeah,
absolutely, and in Australia as well. Yeah,
it's terrible, and it's such a hard issue to solve, particularly small cell lung cancer. So could you talk to us then about why you're specifically looking at this? I mean, as you said, it is one of the hardest types to find a cure for. What drew your attention to this area in particular?
So basically, I was always, I had always an interest in understanding the complex mechanisms of, diseases, basically, especially cancer, but at the molecular level. I wanted to research small cell lung cancer because it's an area of urgency. Basically, it's one of the most aggressive cancers worldwide and in Australia. It has really fewer treatment options compared to other cancers, and it has really, really dismal or poor outcomes. I think the survival rate from that cancer, for small cell lung cancer, is less than 5-year survival rate is less than 7% for patients. So it's you can imagine how bad or how terrible this disease is. And there is not much research is going around for that type of cancer. So I wanted to basically try to make a difference.
Yeah, absolutely. And I think the stats we have here as well is that over 70% of those with this type of lung cancer pass away within 24 months. So
Absolutely.
it's horrific stats, and very much, as you say, needs a focus as well. So we talked about these words, small cell lung cancer. To the lay audience here and to myself, can you explain what small cell lung cancer is compared to normal lung cancer, as we term it?
Sure. So basically, as you said, lung cancer is the most the most important killer, basically, or the lethal, the most lethal cancer worldwide and in Australia. It can be defined on the histology side of things, under the microscope, into two major groups. The first group is non-small cell lung cancer. And these are basically historical names. So they identified small cell lung cancer first, which appears under the microscope as small oat-like cells. That's why it's called small cell lung cancer, right? And then there are kind of lung cancer that is bigger in size under the microscope. So they named it non-small cell lung cancer. Usually, non-small cell lung cancer accounts for 85% of lung cancer patients, and small cell lung cancer is only 15% of lung cancer patients. And as you can imagine, because 85% of lung cancer patients present with non-small cell lung cancer, most of the research is going through, going to treat basically non-small cell lung cancer. That leaves the small cell lung cancer patients without any novel treatments like we see in non-small cell lung cancer area. So that's basically the terminology, why it's called, it's historical terminology, basically. So.
Yeah, thank you for explaining that. Very helpful. A question we had that came in was, can someone have non-small cell lung cancer and then can it change? Is it an evolution, or do you either have one or the other?
Yes, we can see that specifically so there is a type of small cell lung cancer. It's coming from non-small cell lung cancer patients treated with specific inhibitors. Okay. As a side effect of those inhibitors, the tumors evolve to produce a more aggressive subtype, which is the small cell lung cancer. So about 10% of non-small cell lung cancer patients that are treated with EGFR inhibitors, subtype of, one of the treatments for non-small cell lung cancer, they have a potential to develop small cell lung cancer, a more aggressive disease. So the answer is yes. So yeah, it can be.
Yeah, thank you.
Happen, yeah.
And then a question here that's come in about how do you get the different types of lung cancer? And obviously, if we knew that, we'd have a crystal ball. But as far as you know from your research, what are the causes of small cell lung cancer and non-small cell lung cancer?
Yep. It's an excellent question. Basically, the major risk factor for lung cancer in general is smoking. About 80 to 85% of lung cancer patients, they were smokers, previous smokers, secondhand smokers, they had some sort of, exposure to smoking at certain point. What smoking does to our lung cells, it mutates it. So it destructs the DNA, changes the DNA to bring up mutations, right? It depends on what type of mutation. Basically, you get certain type of cancer. So for example, smoking can induce something called EGFR mutations or KRAS mutations, right? KRAS mutations are mainly non-small cell lung cancer. It presents as small cell lung cancer. Non-small cell lung cancer. So smoking, if it mutates KRAS genes, it produces non-small cell lung cancer. In small cell, there are two types of mutations that basically accounts for 90% of patients. So if smoking or any exposure to any chemical induces P53 mutation and RB1 mutations, these mutations will lead to small cell lung cancer. So it depends on the type of mutation in the cancer, which basically governs how the tumor presents at the end. So that's basically it. There are, it's more complex than that, but usually what we see on the genetic levels, there are certain mutations associated with non-small cell lung cancer and certain mutations associated with small cell lung cancer. And in small cell lung cancer, two types of mutations, P53 and RB1, are basically 90% of patients will present with those two mutations.
Thank you. That makes sense. And why is small cell lung cancer so aggressive?
There are so many reasons for that, but one of them is the mutational load in these cells or in these tumors. They can change very, very, very rapidly, basically. And that's why they can proliferate aggressively. They can metastasize aggressively as well. We see a lot of metastasis in patients with small cell lung cancer. And also they have a really bad response to chemotherapy. So at the beginning, patients, the main treatments for small cell lung cancer is chemotherapy, right? So when patients undergo chemotherapy, the response rate really high. So 90% of patients initially respond to chemotherapy. But after 6 months or so, about 90% of patients will undergo relapse, which we call chemoresistance. So tumors, because of the mutational load in these tumors, they can mutate, they can evade the chemotherapy, and they can come, more aggressive than the initial tumor.
Yeah, thank you. Just going to pause there. We've had a couple of new people join. Welcome to our Lunch and Learn. We're talking about the crucial work on small cell lung cancer, a very lethal form of lung cancer, where over 70% of people that have this pass away, sadly, within 24 months. So Dr. Mohamed, we talked in the opening spiel here about you creating an off switch, which is incredible given how aggressive this is, and obviously the fatality rate too. Could you explain more about how you discovered this and
Sure.
what this off switch is?
Yep. So we started this work in the non-small cell lung cancer area during my PhD. And that was basically the thought-provoking idea is to try and test what we've done on the non-small cell lung cancer in small cell lung cancer. So just to make things clearer, and I don't know if you see this. So basically consider this as a tumor cell, right? It has proteins on the cell membrane, and these proteins are important for signaling. So that tumor cells can talk to each other, basically. The proteins we are interested in, it's called proteases, and consider these as scissors, molecular scissors on the molecular level. So the protein I'm interested in is called ADAM17, and it works on the cell membrane, on the surface of the cell. And that scissor cleaves off another membrane proteins, right? So once this cleaved, it goes to another cell and basically activate or signal to this cell to do something. And that something is proliferation usually, or metastasis. So tumor cells use ADAM17 or use proteases on the cell membrane to propagate the signal from one cell to another so that you get a tumor at the end. And that tumor can proliferate aggressively and then also can metastasize to distant places in the body. So the idea is if we can block that protease from releasing these signals that basically will lower the proliferation rate of these tumors and also lower the metastasis rate of these tumors. We also look at the proteins that are released in the bloodstream as biomarkers of the tumor. So if we can detect these proteins early on, and we know it's increased in that specific type of lung cancer, we can basically use these proteins to detect or to use it as biomarkers to detect lung cancer early on, or that specific type of lung cancer early on, before it metastasizes or before it gets bigger. So that's basically what we do in the lab.
Lovely, thank you, and thank you for showing us it. Demonstration always helps us understand. So you've had this concept and you're seeing that it could work. Where to now? What do? You've kind of got this discovery. How do you get it from here to eventually one day assisting patients that have small cell lung cancer?
Great question, Nikki. So basically, there are, with the Cure Cancer Funds, at the moment, we are working on 3 different topics, basically. So the first one, with Cure Cancer Funds, we could form a collaboration with researchers at University of South Australia. They have a proteomics facility. Proteomics mean understanding the proteins at the molecular level. So we collect the samples and then we send it to the proteomics lab, and we can basically understand through the analysis of the proteins in our samples how these proteases work and what the biomarkers we are interested in, how they work, how they interact with each other. So we can understand that on the molecular level. So that's one front we are working on. The second one, it's actually a challenge in small cell lung cancer because most of the patients with small cell lung cancer, they don't do resection. Like there is no, the resection is not a treatment for small cell lung cancer. In non-small cell lung cancer, you get resection tissues from patients, but it doesn't happen in small cell lung cancer. So what we've done, which is really exciting for us at the moment, we formed a collaboration with thoracic surgeons at Royal Adelaide Hospital. Ethics has been approved in the last 2 weeks, and we started basically collecting tissues from patients that basically undergo a diagnostic procedure. So to diagnose small cell lung cancer, they usually undergo a biopsy collection procedure. And with the patient consent, we can, instead of collecting 2-3 biopsies from the tumor, we can extend that to 5 or 6 biopsies, and we take 2 or 3 of these biopsies for research. What we can do with these, it's really valuable materials for us. So we can grow these in the lab to make it bigger and to understand more, how the tumor is forming. And we can send it to proteomics as well, to the proteomics team, to analyze what's happening on the molecular level. And by this, it's invaluable knowledge for us to know how the proteins are working in these tumors. The third front, we're working on at the moment is we're trying to develop an inhibitor, basically, that we can use down the track in the clinic to inhibit the protease of interest for us. Mm-hmm. So we are having some discussions with pharmaceutical companies with the best way, these discussions will lead to the best way on how to target that protease, basically, down the track for future clinical interventions.
It's very exciting, and as you say, so needed as well. So congratulations on this discovery, and let's hope that this can speed up the process so that you can get it into the clinic for these patients as well. I have a question here about actually types of causes of lung cancer. I don't know if you're able to answer that, but is non-smoking lung cancer, does that also show signs of small cell lung cancer, or is what you're seeing generally driven by people that have been smoking?
Mostly in small cell lung cancer specifically, most of the patients are smokers or secondhand smokers. Mostly, yes. In non-small cell lung cancer, it's a different story. 80% of patients usually smokers, but you get about 20%. They call it never smokers, and it's purely genetics. You know, in the family there is one gene that is mutated, and that increases the risk of having lung cancer down the track during your lifetime, basically. Um, so yeah, in small cell lung cancer, it's mainly associated with smoking.
Understand. Lovely. Thank you for answering that.
No worries.
So for a day-to-day basis, can you explain to the audience, you know, what does your typical, if there's such a thing, typical day look like?
Oh, one of the great things I like about my work is it's never day one is never exactly the same as day two. That's challenging, and also it's rewarding at the end. You never get bored. So basically, I can have meetings in the morning with our team, have meetings with my mentors, basically, to see how the project is going, do experiments in the lab in the afternoon, and then go home, write some grants, write papers, analyze data, and the procedure goes basically, the cycle basically repeats itself. Second day, I could have full day of experiments in the lab. I have a big experiment. And then the day after, I can, reserve that day basically to write grants or write papers. So every day is different from the day before, basically. But as a researcher, it's always busy because you do multiple jobs at once. So you need to keep up with what's happening in the research area. You need to have meetings with your team members. You need to do experiments. You need to analyze them. So yeah, lots of things. And you need to give talks at conferences and learn on lunch with Nikki. So.
And we appreciate your valuable time, absolutely.
Thanks.
A question here from a person called Maha, who wanted to know, what are your biggest challenges from trying to turn a discovery into an actual treatment?
I would say funding.
Mm.
Yeah, funding in our research is basically essential. Without funding, we don't have lab resources, we don't have collaborations, we don't do experiments. And as you know, Nikki, funding is basically scarce, unfortunately. So with funding, we can transform any promising ideas into basically meaningful solutions for technically any disease we have, basically, to improve the lives of our patients. So that's the most challenging situation we face in research in general, is funding.
Yeah, absolutely. And obviously this is why we exist as an organization, but the funding that you have today is absolutely down to our generous donors and supporters. So a big thank you to them. You're looking at some hopefully incredible results from a very lethal type of cancer. So hopefully this can start to make a difference in people's lives as well. On the flip side, what motivates you? You know, that it is challenging. You have to essentially run your own small business. You have to secure funding. Every day is different, as you put it. But what motivates you to keep going?
It's a tricky question. Because I'm usually self-motivated. I like to wake up. Let's see what's new in our research and just do it, basically. Knowing at the end that you might change the life of someone, that's a big, for me at least, it's a big accomplishment. To be able to detect this aggressive tumor at early stage and change the lives of these patients, or extend, the quality of life for these patients by some years, basically, that's a great achievement for me. So I'm working towards that, basically. So that keeps me going.
Absolutely, and that's a huge goal to achieve, isn't it? So thank you. We've got about 15 minutes left with the lunch and learn for Dr. Saad. So if you want to pop another question, we're getting loads of them, so thank you. If you want to pop another question into the chat, I'll weave that into the conversation as well. I have a question here from an audience member now that's asking about this whole off switch that you had developed. Do you think this is just for small cell lung cancer, or can it actually be used for other types of cancer as well?
It's a good question. We started this work in non-small cell lung cancer, and there is a specific type in non-small cell lung cancer that we can use that switch to inhibit the proliferation of the tumors. So we can technically use it in other types of lung cancer. It depends on how upregulated that protein is. So we see it highly upregulated in small cell lung cancer. That's why we are focused now in small cell lung cancer. And we have the tools basically to be able to understand how this protein functions in every subtype of lung cancer. So that's basically an easy task, I would say. If we can know basically if that protein will affect or is important in lung cancer subtype or not. But at the moment, we're focusing on small cell lung cancer because, as we said, it's the most aggressive subtype, and not many treatment options are available for these patients. So if we can basically identify that protein as a therapeutic target for these patients, we can expand basically these discoveries to other subtypes of lung cancer that share the importance of that protein basically in their proliferation or yeah.
Lovely, thank you. I also have a question here about metastasis. You talk about how it proliferates very quickly. It's a very aggressive form of cancer. Have you seen, and again, I know, appreciate you're not a clinician, but have you seen it metastasizing into other areas of the body, or does this type of cancer generally just stay within the lung region?
No, it's highly metastatic, unfortunately. We see it in patients, because we have discussions with the patients that donate the tissues for us. So most of the time we see metastasis, usually brain, lymph nodes, liver as well, adrenal gland. And we also have preclinical models to model small cell lung cancer in the lab as well. And we also see metastasis in these preclinical models. So we see the tumors goes to other areas in the body, basically.
Okay, so this treatment could not only help or slow down the proliferation, hopefully cure it, but also prevent the metastasis into other forms of cancer as well.
Yes, definitely. Yeah, we
Lovely.
hope so. Yeah.
Thank you. Question here from the audience about what inspired you. How did you come across this protein and the proliferation, and how did you know to go from where you were to where you are now?
These are 10 years of my life, I think. So it's all, to be honest, it's all about funding and collaboration. So the idea was we were attending a conference and we showed some data on that protein, in non-small cell lung cancer. And then we had a discussion with one of the professors in Germany. They had a preclinical model that don't have that protein specifically. And then all of a sudden we had this big collaboration with them to understand how that protein is working. And then we published a really nice paper on that project, basically showing that that protein is really important in non-small cell lung cancer. And then once I moved to University of Adelaide, we had this collaboration with the proteomics people and surgeons at the Royal Adelaide Hospital. So if you have funding and you have good people or good collaborations around you, ideas basically bounce, and you can do whatever you like in research. So yeah.
Lovely. And a question here about the collaborations. Are you looking at samples? You mentioned the University of Adelaide and the hospital in Adelaide. Are you looking at samples from overseas as well? And if you are, do you see any difference in geography from patients, or is it typically the same trend in metastasis in patients that you've seen the samples from?
It's a great question. We have some collaborations with people in Germany that gives us some. It's the tumor, the biopsies we get from small cell lung cancer is tiny.
Mhm.
So we had some tissues from Germany, from about 90 patients. And basically, it's exactly the same genetic material, or genetic mutations that we see in all patients of small cell lung cancer. I don't think there is any differences in geography.
Yeah.
Smoking is smoking everywhere.
Yeah.
Mutations are mutations everywhere. And pathology of these tumors are same everywhere. So I don't think we saw, and the overexpression of our protein of interest is across the board as well. So we can technically use inhibitors for that protein across all patients or samples of small cell lung cancer. So yeah.
Very interesting. That's the first time we've heard that. Often when we talk to researchers, they say the ethnicity of someone or location, it causes differences, but it's interesting to learn that they're all very similar.
90% of small cell lung cancer patients, no matter where they are, will have P53 mutations and RB1 mutations, and these are the most significant mutations we see in small cell lung cancer. So there is no other other options.
Yeah.
Those two mutations will develop small cell lung cancer.
Yeah. A question here about how you can reduce the risk of this. Obviously, we talked about the impact of smoking. Is any of this genetic from the small cell lung cancer? If so, I'm not sure there's much you can do, but is there any way that you can reduce your risk from getting this type of cancer?
Genetically, I don't think so. But as you said, the major risk factor for lung cancer in general is smoking. So my advice, my single advice, or the only advice, stay away from smoking as a first-hand smoker or second-hand smoker. Also try to stay away from air pollutants as well, whether it's occupational or from any source basically of air pollution, that basically a risk factor also for lung cancer. So smoking is the only risk factor, modifiable risk factor, that you can basically or easily stop to reduce the risk of having lung cancer.
A very strong message there, isn't it? Thank you for that. I've got a question that's come in here about why is lung cancer research on the whole so far behind other types of cancer research?
Good question. The tumor is very elusive. That's how I understand it, how I think about it. If there is a lot of research happening in non-small cell lung cancer, especially with targeted and immunotherapy on the horizon at the moment, and it's been used in the clinics. But unfortunately, the tumor is very, very clever. So once you hit it with one inhibitor or one treatment, it comes back again stronger, more mutated. And basically, that's our job at the moment, to understand how the tumor evolves and how can we, target these tumors basically until we kill it, if we can. So that tumor resistance in lung cancer is a major problem, and part of our job is to try and understand how resistance is developing in these tumors, so we can be better equipped basically to inhibit these tumors. So that's I hope that answers the question. I think tumor resistance is the major factor in this in this problem.
I believe that often when people do have lung cancer, it's diagnosed late as well in a lot of people.
Unfortunately,
Yeah.
late stage, yeah, unfortunately,
Yeah,
yeah.
lovely. Thank you. A question here about advice for early career cancer researchers. Obviously, that's solely what we fund, and it's an honor because we fund innovation like the work that you're working on now, different ways of doing and treating cancer. What advice do you have for anyone here on the call that might want to get into research or might want to change their career and move into research?
I would say resilience is one thing you should have before entering the realm of research, because there is a lot of downs in, experiment doesn't work, funding you didn't get, paper rejected, a lot of downs, but there are a lot of ups as well. When Nikki called me to tell me that I've got the grant, so that was a really a massive point, or a good day basically I had in research. So resilience, I would say, and also mentorship. That's really, really important. It's really important to be surrounded by people that motivate you, guide you through the right path, basically. So all we do is basically discussions about our work. You can get one idea from here, one idea from there, to advance your work. So without mentorship, we can't move basically forward. So yeah, those are my main 2 points, I think.
Thank you. And I must say that it is my most favorite part of the job every year when I get to call researchers and tell them that we're funding them. It's an incredible honor, thanks to our generous donors. But it's just so exciting, and we look at the work you're doing, and it is cutting edge, and it is solving a very big problem. So it's great to be able to have these funds to channel in your direction as well. Couple of last questions before we wrap up. What are your hopes for lung cancer or small cell lung cancer? Where do you want this to go in the next 2 to 5 years?
Yeah. So basically, one of the aims we're working on is to identify a protein that we can inhibit, that reduces the proliferation and metastasis of this tumor. Also try to develop an inhibitor for that protein that we can use in preclinical work first, and then we expand it to the clinic, hopefully in the future. And the third thing is to identify early biomarkers, if there is, that we can basically use to early detect small cell lung cancer, because as you said, it's mostly detected at late stages, and those late stages, the treatment options for those late stages are really scarce, basically. So
Yeah, lovely. Thank you. And then the final point or message, do you have anything to say? We've got a bunch of generous donors on the call now. They will view us afterwards on the YouTube as well. So do you have a message to those that have supported us and also your project?
Absolutely. Well, first, thank you very much for your generous donations. Without you, I wouldn't be here today. I wouldn't be discussing this exciting work. I wouldn't be collaborating with people all around the world, basically, to advance this project. I think, like I said, funding is essential for us. Every dollar makes a difference, basically. It supports our lab resources. It's supports our team expertise, supports forming collaborations with people to basically understand this disease and make discoveries. And without your donations, we wouldn't be able to do that, unfortunately. So thank you very much, and I hope you keep donating for the good cause.
Yeah, they're very generous, and it's been an honor. I think over the last 57 years, we've funded over 550 new research projects, which is incredible, but it's also a testament to the brilliant researchers that we have here in Australia and the work they're doing. So thank you for sharing your valuable time with us.
you.
We do have a profile page on our website about Dr. Mohamed, so you can go there to read more about his work. If you do have any other questions that we haven't been able to fit in, please send those to info@curecancer.com.au and we'll pass those on to Dr. Mohamed. Thank you for your work. Please thank your team as well. It's an honor to hear about your work and I wish you all the best. And hopefully we can get the concept into reality very soon as well. Thank you.
Thank you.
Thank you all for joining us in today's Lunch and Learn. Please join me. We will be next talking to Dr. Kelly Mushmore, who is a postdoctoral research fellow at the Olivia Newton-John Centre. And we'll be having a great discussion with her on the 28th of November at the same time, talking about her groundbreaking work on another very deadly type of cancer, and that's triple negative breast cancer. So thanks for your time today. This conversation will be sent to you via email next week, and you'll also find it on our YouTube channel as well. Wish you all the best, and I'll see you on the 28th. Have a good afternoon. Thank you.
Thanks, Nikki. See you.